Thank you very much for this nice introduction and for the invitation. It's always a pleasure to come to Canada, and more importantly, it's really interesting to learn from you how you deal with the challenges in the field, how to treat patients with hepatitis C. Obviously, there are differences between Switzerland and Canada in the problems, but it's very important that we can learn from each other. The title that I have been given by Jordan, as you see, is a kind of a difficult title. I think during the last 20 years, many of us have tried to understand the interaction of the virus with the immune system. And I think we have made some important progress in understanding this important interaction. Probably, we all feel a little bit threatened now by the rapid development of these highly effective direct antiviral treatments. We don't know how long we are going to be funded still for this kind of research. So it might also be one of the last opportunities to talk about the innate immune system in the context of hepatitis C. So thank you, Jordan, for giving me this opportunity. So I would like to start with a few slides that highlight the role of the innate immune response in interferon-based treatments, and then to put this in a perspective to the role of the immune response in the area of interferon-free treatments. So I think. So if you do a liver biopsy in patients with chronic hepatitis C, and you stain this biopsy for interferon-stimulated genes, and this is an example of such a gene, USP18, you can find these two extreme situations. On the left side, you have a very strong expression of this protein in basically 100% of the liver cells, and on the right side, you see you don't detect the protein at all. And this expression of interferon-stimulated genes is not normally distributed in a population of patients. So it's not the maximum is not at the medium, but it's a U-shaped distribution. And we know that it has to do with response to interferon-based treatments. And you see here this distribution, which gives you the probability of response to pegylated interferon and ribavirin from 0 to 1. And you can see there are a large group of patients with a very high probability on the right side, which don't have this USP18 or don't have interferon-stimulated gene expression. And then on the very left side of this graph, you have the patients with induced interferon-stimulated genes in the liver, and they have a very low chance of responding. So this is a U-shaped distribution, and we don't understand exactly what makes the decision in an individual patient between having an activated interferon system in the liver or not having any activation of the interferon system in the liver. An important clue comes from investigations that linked the genetic variations in the interleukin-28B locus, the interferon lambda 3 locus. These are identical locuses. With hepatic gene expression, and you don't have to read here all the details, but what you can see is that in patients that have an IL-28B T allele, we have a strong expression of interferon-stimulated genes, and in patients that have that are homozygous for the C allele, we have no expression of these genes. And of course, we all know this seminal work 3-4 years ago from 4 different groups found independently. Genome-wide association studies identified these genetic variants in the interferon lambda 3 locus to be highly significantly associated with response to treatment with pegylated interferon and ribavirin. More recently, it was realized that there is probably a fourth interferon lambda gene, interferon lambda 4, and this fourth gene is only transcribed from the delta G allele, and this is the ancestral allele. So if you go back in evolution, interferon lambda 4 was always there, but in humans there was an insertion deletion, so two T's replaced a G, and this led to a mutation that is no longer able to produce interferon lambda 4. So there is increasing evidence that interferon lambda 4 is playing an important role here in the reaction of the immune system to hepatitis C virus. At the moment, the most difficult problem with the concept is that it has not been reproducibly detected in human liver biopsies in interferon lambda 4. But that might be a matter of time till there is more evidence. So to summarize this part, I think what we have learned from a huge amount of work in this area is that genetic variations or variants at the interferon lambda 3 or IL-28B gene locus, probably involving the production or non-production of interferon lambda 4, are associated with induction of interferon-stimulated genes in the liver. And this is just one example that I show you here, USB18 in this immunostaining. And those patients that have this activation of the endogenous interferon system, they cannot respond to treatments with pegylated interferon alpha and ribavirin, whereas those that have no such pre-activation, they are very, very well responding and usually have a sustained virological response. So now let me come to the role of this innate immune answer in interferon-free treatments. And there is some evidence that the IL-28B genotype is also associated with decline of viral load in interferon-free treatments. And I found two studies in the literature that I cited you here, and I show you the data of that. So this is the first one. You can see that depending on the IL-28B genotype, CC, TT, or TC, you have a significantly different decrease in viral load during treatment. And the same has been found here in this other study that has been published very recently, sofosbuvir and ribavirin. Again, there is a difference in the decrease of viral load during treatment. There has even been a study published that showed that the IL-28B genotype is associated with SVR. And I can show you the data here. If you concentrate on the lowest two rows of this graph, you can see that IL-28B-CC, this is the favorable genotype. If you look there at the sustained virological response rates, they are around 67 to 80%. And then if you go down a row to the non-CC, you see that it's about 15 to 20% less in each case. So this is a study that shows dependence of SVR on IL-28B genotype. But of course you also realize that these 67 or 55% in this time are rather low numbers. But there is also evidence to the contrary, and this is one study that has also been published very recently that had found no association of the decline of viral load with IL-28B genotype. And then there are a number of studies published last year and this year that showed that there is no association of SVR with IL-28B genotype, and I have summarized them here. Many of you probably know these studies. And then I have taken here a slide from clinical care option hepatitis slide set that was curated by Forster, Zoitzam, and Nelson. and covered the AASLD 2013 talks in regard to interferon-free therapies. And I don't expect you to go into all these details, but you see here the numbers. They are approaching 100%, and we have heard that also this morning. And I think it's pretty clear that if you have 100% response, IL-28B genotype doesn't matter anymore. I mean, you cannot have more than 100% response. So, to summarize this part, I think what we can say is that the IL-28B genotype, interferon lambda 4, and the innate or the immune response in general to HCV have no significant impact on virological response in interferon-free combination treatments with highly potent direct antiviral agents. And this is probably because if you inhibit replication of the virus strongly enough, it just disappears. But this would be something that my chair, Mike Houghton, is probably much more knowledgeable about. It seems that the virus has no reservoir where it can be in an inactive form. It has to replicate all the time, and if you stop replication, regardless of the presence or absence of an immune answer, you probably just cure the patient. That's my interpretation of the current evidence that we have in the field. Okay, I basically could stop here, but I have some more minutes to go. And so I thought to come back a little bit to the real life situation, and then I made a third part of this talk, which is entitled "The Role of the Innate Immune System in the Era of Expensive Interferon-Free Therapies." And I mean, I think we all realize that interferons probably will not go away so soon in the treatment of hepatitis C. And this is just a calculation based on our numbers in Switzerland that can highlight a little bit why I think interferons will stay in the armamentarium for treatments of hepatitis C for some time at least. So if you look at these numbers, we think of the interferon-free treatments will be about 100,000, maybe even more per person. And in Switzerland, the entire money spent per year on drugs, on all drugs in Switzerland, is $5,500 million. And you see the calculation. To treat all the patients infected in Switzerland, we would need about $5,000 to $9,000 million. And even if we split this over the next 10 years, it would still be about 10% of the entire budget just for one disease over the next 10 years. And I can tell you, Switzerland is a rich country, but it will just not going to happen in Switzerland. It's not possible to increase the budget by 10% in the next years. So this is the background of a discussion that I summarize here. You know, what are the strategies for doctors that have to deal with this hepatitis C epidemic? Of course, I think the first thing we will do, we will use these expensive treatments for those who are most in need. That's probably going to be a reaction all over the world. And then we will hope that the prices come down because there will be more drugs, and hopefully this will reduce the number of the cost of the drugs. And then there is another strategy that we can think of, and this is we could try to optimize the current treatments or to optimize treatments with current available drugs with pegylated interferon alpha and maybe lambda. Why do I think it could be worthwhile to optimize interferon alpha treatments? One piece of evidence I have summarized here, and we all know that interferon alpha is highly effective in acute hepatitis C, and there are I listed some studies. There are more studies out there. It is effective also in monotherapy. You don't need a ribavirin, and it can be effective in short treatments of 12 weeks. This is the most recent publication from Italy showed that there is no difference between 24 weeks and 12 weeks of treatment with pegylated interferon alpha 2b monotherapy for acute hepatitis C. So 12 weeks of interferon alpha in acute hepatitis has a very high SVR rate. And I would like to stress this also, toxicity in all these acute hepatitis C trials seems to be quite low, much lower than what we have in this chronic treatment. And I think this is also a very interesting slide, over 13 years old now, from one of the first trials with treatments of acute hepatitis C. And what you see here is that interferons also work very rapidly. After 4 weeks of treatment, there is basically 90% of the patients had an undetectable viral load. So it is also a very rapid acting drug in this setting of acute hepatitis C. Now, what is so different between acute hepatitis C and chronic hepatitis C? And we had the opportunity to analyze liver biopsies from patients with acute hepatitis C, and we did a transcriptome analysis and compared the gene expression profiles in acute hepatitis C patients with those of chronic hepatitis C patients. What you can see in red are the acute hepatitis C patients. In yellow, these are chronic hepatitis C patients that were responders to interferon ribavirin combination treatment. So those are the patients who have no pre-activated interferon system in the liver. And in blue, you see the non-responders. So those are the patients that have hundreds of genes, interferon-stimulated genes induced in the liver. And to make a long story short, the take-home message here is that in acute hepatitis C, we have an interferon gamma gene signature that is upregulated, whereas in chronic hepatitis C non-responders, we have an interferon alpha gene signature that is upregulated. There is a difference in the pattern of ISG upregulation between these. We know from work in chimpanzees that at the very beginning of a hepatitis C virus infection, there is also an interferon alpha a gene signature upregulated, shown here in the first 6 weeks. But then we have an influx of the cellular immune system, and interferon gamma starts to dominate the field. Interferon alpha induced genes go down. And then if you have a persistent infection, interferon gamma genes go down again in the liver. And what we have in the chronic phase is either no interferon-stimulated gene induction, shown in white here, or then in green, interferon alpha-stimulated gene inductions. Now, I showed you at the beginning, one of these genes that is upregulated in chronic hepatitis C non-responders is USP18, and USP18 happens to be a very strong inhibitor of interferon alpha signaling. It inhibits the interferon alpha receptor. And what we found here when we looked in these samples of acute hepatitis C, you see at the bottom D, you see the control, no expression of USP18. Acute hepatitis C, no expression of USP18. And I have to say, this is in a sample that has hundreds of genes upregulated, interferon gamma stimulated genes, but not USP18. And then you have the non-responder patient that I've already shown you with a very strong induction. So to conclude this, we think the reason why you have such a good response to interferon alpha in acute hepatitis C is because you don't have the upregulation of inhibitors of interferon alpha signaling. One of them is certainly USP18. And in chronic hepatitis C non-responders, the reason that you don't respond is that you have a very strong inhibition of interferon alpha signaling in these cells. Now there is a perspective to that, and this comes from work that has been published by both Lanford in 2010. And he treated chimpanzees with anti-miR-122, and thereby inhibiting viral replication. and you can see the viral load in this dotted line, and you can see ISG expression in green and in red. So interferon regulates genes in both directions. It upregulates genes and it downregulates genes, and if you concentrate on the green ones, these are genes that are upregulated by interferon, and if the viral load goes down, all these genes go down also. So what you could speculate about is that if you use a direct-acting antiviral, you reduce the viral load, you reverse this activation of the interferon system in the liver, and you might make patients responsive to interferon again, similar to a situation that we have in acute hepatitis C. Optimizing treatments with speculated interferon alpha. So this was the first point of this list that I discussed now, and I have listed some other options that I will very shortly discuss also. So we could try to improve patient selection, that we select patients that have a very high chance to be cured with interferon. Maybe even in shorter duration treatments. I will talk a little bit about dosing intervals. We have recently investigated interferon signaling during the first week after pegylated interferon alpha in liver biopsies that we took during the first week of treatment. I will show you some data from that. And it looks like we are not using interferon very intelligently at the moment. And then I will also like to discuss some aspects of interferon, pegylated interferon lambda. So this is from this paper where we looked at interferon signaling in biopsies taken after 4 hours, 16 hours, 48 hours, 96 hours, and 144 hours after the first injection of pegylated interferon in patients with chronic hepatitis C. And what we look here, or what we assess here, is interferon-stimulated translocation of STAT1. STAT1 is the most important signaling molecule in interferon signaling, and it goes into the nucleus and induces interferon-stimulated genes. And you see that there is a very strong nuclear translocation at 4 hours and 16 hours, but it then rapidly goes down. And you realize that this is in presence of very high serum concentrations of pegylated interferon alpha. So you have pegylated interferon alpha at day 3, 4, 5, 6, and 7 in the blood, but it does not stimulate interferon signaling in liver cells. It's in the blood, but it doesn't do anything in the liver. So it's basically a waste of, I don't know what, but it doesn't do anything in the liver, this pegylated interferon lambda. Now, one of the proteins that is induced by the activation of the JAK-STAT pathway is again this USB18. And you see at 4 hours there is no expression, but already at 16 hours there is strong expression that is maintained over the entire interval. So what you do when you inject pegylated interferon alpha into a patient, you induce the negative regulator of interferon signaling, which shuts down interferon signaling from day 3 until the rest of the dosing interval. Not very intelligent use of a drug, if you think about it, but that's what we do since more than 10 years. Now to the last part, the interferon lambda story. We became interested in, mostly because of this IL-28B and interferon lambda 4 progress that has been made. We became interested again in the interferon lambda receptor, or the interferon lambda system in the liver. And we looked at interferon lambda 1 receptor 1 expression in liver. And interferon lambda 1 receptor is required for signaling or for interferon lambda to signal into a hepatocyte. And what you can see here is that we found a correlation of the receptor expression with the IL-28B genotype. So patients with the favorable genotype CC have a lower expression of interferon lambda receptor than those with the unfavorable genotype. And I don't show you all the data, but we know that the interferon lambda receptor is an interferon stimulated gene itself. So those that have a pre-activated interferon system in the liver, they also have a high expression of the interferon lambda receptor. And not surprisingly, it correlates with response and non-response to pegylated interferon alpha ribavirin treatments, that is shown on the right side. Those who are pre-activated and happen to have also a high interferon lambda receptor, they are non-responders to interferon alpha, to peg alpha and ribavirin. We then took liver biopsies from patients, and immediately after we obtained them, we put a piece of it in a test tube with interferon alpha and in one with interferon lambda, and we measured the interferon lambda receptor expression. And what I show you here is a set of 9 biopsies. All of them have very low interferon lambda receptor expression. So they are not pre-activated. They are patients that will respond quite well to interferon alpha and ribavirin. And that, I think you can see that by eye, the interferon alpha line, you have these red dots. This is again phosphorylated STAT1 that has translocated into the nucleus. So they respond all quite well. But because the receptor to interferon lambda is not expressed very high, they don't have a good response to lambda. And then we have a set of 9 biopsies that have a high expression of the interferon lambda receptor, and you probably can you see it immediately that, of course, if the receptor is expressed, they have a nice response to interferon lambda stimulation, but the interferon alpha stimulation is in many of the samples is not occurring, because these are, as I said, pre-activated patients. And this is more quantitatively shown here. You can see that these samples are now ordered in increasing expression levels of the interferon lambda receptors from the left to the right. So at the left there is a very low expression of the interferon lambda receptor, and you see the yellow bars, there is very little stimulation of STAT1 by interferon lambda, but a very good response to alpha, and the other way around. So there is kind of a reciprocal responsiveness to these 2 types of interferons, at least ex vivo, when we do this with biopsies treated immediately ex vivo. So this is I'm nearly finished. I just wanted to give you some food for thoughts, for ideas, maybe for research. Could we could we think of some intelligent combinations of DAAs with interferons? Could we decrease costs by using some very potent DAAs for a shorter time and then coming in with the traditional interferons with maybe very, very good response rates? This is a hypothesis, but it's a testable hypothesis and might really save costs for a health economy. I think in the area of patient selection, also with a little bit of creativity, I think we could do better than just treat everybody with the Perfectovir, which is too expensive. And the dosing intervals, you know, I think we are not using interferons very intelligently at the moment. From what we see in acute hepatitis C, and also if you think about the spontaneous clearance situation in acute hepatitis C, where about 30 to 40% of the patients, they clear spontaneously, and what is doing the job is also interferon. This is very clear, and those patients have very little side effects. So obviously our body is using the interferons much more intelligently than we are doing. We are just giving probably too much at the wrong time, maybe also the wrong type, I don't know. But you know, maybe the speculation is that if we would know more about it, we might be using these drugs more intelligently, or we might be able to use them more intelligently. And then the last point, one of the ideas could be that you use pegylated interferon alpha and lambda together. With the lambda, you would treat those patients that have a pre-activated system and are not responsive to interferon alpha. And with the alpha, you would treat those that respond to alpha and have no pre-activated interferon system. So to conclude, the role of the innate immune response in HCV in the era of interferon-free therapy, I think in case of available and affordable all oral therapies with highly potent DAAs, there is going to be a declining interest in immune responses to HCV. I think you just don't need them. You can cure the patients without an immune system with these drugs, but that's my personal hypothesis. That this is not proven, of course. I think if these treatments continue to be very expensive or too expensive for many patients, we might have the obligation to further explore the possibility of using available treatments that are cheaper and use them more intelligently, and maybe achieve also very good results for a large group of patients that otherwise would not be treated because of the expenses of the DAAs. And with that, I would like to close. Thank you for your attention, and I would like to thank all my collaborators and friends and group members that have helped me in the last years. Thank you.
Thank you very much, Markus. That was a fantastic talk. It was so interesting. We'll just allow 2 questions. You're dead on time, Markus, but yeah.
Sure. Thanks, Markus, for coming, and thanks for the great talk as usual. Just a question about lambda. I guess 2 questions about the PEG interferons in general. One is, you've told us that we're using them poorly, but the response rates still are much better than standard interferon. So I guess that's one question. Do you have any kind of explanation for that? And the second thing is, in the small studies that have been done with lambda, PEG lambda compared to PEG alpha, the response rates were quite similar, suggesting that those patients with pre-activated ISGs still don't respond to lambda despite what we think they might.
Mm-hmm. So to the first question, so I couldn't show you the data, but we also had a chance to, in a collaboration with Daryl Lau, we got her transcriptome data that she had 24 hours after interferon alpha, conventional interferon alpha. And we looked at the transcriptome of alpha versus PEG alpha. And what we found is that PEG alpha induces an additional set of genes that is mostly involved with cellular immune response. So our hypothesis is, or our conclusion is, that probably the difference in efficacy comes from a better efficacy of PEG alpha for the cellular immune response. And interestingly, if you look very exactly at this immunohistochemistry, even if you go down to 5, 6, 7 days in this first dosing interval, you still have nuclear p-STAT1, but it's not in hepatocytes, it's in non-parenchymal cells. And maybe these are immune cells that are still stimulated by circulating levels of PEG alpha, but not in case of conventional interferon alpha. That's our hypothesis or our thing. And then for the PEG lambda, this is also interesting. We don't have any data. This is pure speculation. But if we measure the lambda receptor in non-infected patients, it's very low. If we measure, and if we then compare with hepatitis C infected patients, there is clearly an induction of the interferon lambda receptor. Now, if you reduce the viral load, you will reduce the expression of the receptor. And it's very well possible that successful treatment with interferon lambda reduces the receptor level and then makes it more and more ineffective the longer you treat. So that's why I have this idea of combining these two things might be an idea to get better response rates. But this is also purely speculative.
Thanks.
But we are allowed to speculate, aren't we?
I'm sorry, Steve. We're going to have to move on. Thank you very much, Markus, for that very stimulating talk.